Hello, everyone. Imagine your body as a vast, intricate communication system. The messages, chemical signals such as hormones, neurotransmitters, and of course, drugs. The receivers of these messages are specialized proteins called receptors. So in this video, we'll explore what receptors are, how does receptor signaling work, and the major receptor theories that form the foundation of pharmacology. Let's begin. So, what is a receptor? Let's start with definition. A receptor is a specialized macromolecule, usually a protein, located either on the surface of a cell membrane or within the cell itself, in the cytoplasm or nucleus. Its main job is to recognize and bind to specific molecules, such as drugs or endogenous ligands, like hormones or neurotransmitters. Once the receptor binds to its ligand, this interaction triggers a series of events inside the cell, ultimately leading to a biological response. This process, known as signal transduction, converts an external chemical signal into a physiological effect within the body. So, why are receptors so important? Almost every drug you take works by interacting with these receptors. For example, agonist drugs mimic the action of natural substances and activate receptors, while antagonist drugs block receptors so that natural substances can't activate them. Some drugs even tweak how receptors respond, acting as modulators. Understanding receptors helps explain why some drugs work, why some don't, and why side effects occur. The type, number, and distribution of receptors in your body determine the effects and selectivity of drugs. For example, imagine two patients each given the same dose of a pain medication. One feels strong relief, while the other experiences little effect and more side effects. This can happen because the second patient might have fewer or differently distributed receptors for that drug, making their body respond differently. Recognizing these receptor differences is key for clinicians to tailor treatments to each patient's unique biology. Now, let's see how to understand the molecular nature of receptors, starting with membrane receptors. Most receptors are proteins embedded in the cell membrane. Think of them as gatekeepers, constantly monitoring the external environment and deciding how the cell should respond. They detect signals from molecules like hormones, neurotransmitters, and drugs, and convert these signals into cellular actions. These membrane-bound receptors are essential for rapid communication, especially in systems like the nervous system. Next, intracellular receptors. Some receptors are located inside the cell, either in the cytoplasm or the nucleus. These are known as intracellular receptors, and they typically respond to lipid-soluble molecules, such as steroid hormones, that can easily cross the cell membrane. Once the ligand binds, these receptors often act as transcription factors, directly influencing gene expression. So instead of producing a rapid response, they create slower but long-lasting effects, moving to the structure of receptors. Regardless of where they are located, receptors have two key functional regions. They have, first, the ligand binding domain. This is the specific part of the receptor responsible for recognizing and binding a ligand. A ligand can be a drug, hormone, neurotransmitter, or any endogenous signaling molecule. The structure of this domain determines which molecules can bind to the receptor, thereby conferring specificity and selectivity. Because receptors have unique three-dimensional shapes, only molecules with compatible chemical structures can interact effectively with them. This is often compared to a lock-and-key mechanism, in which only the correct key fits into the lock. This selectivity is extremely important in pharmacology because it allows drugs to target specific receptors and produce desired therapeutic effects while minimizing unwanted side effects. For example, beta-1 receptors in the heart respond mainly to norepinephrine, insulin receptors specifically bind insulin, and opioid receptors selectively interact with opioid drugs. Next is the effector domain. Once the ligand binds to the receptor, the effector domain initiates the cellular response. Binding alone is not enough. The receptor must transmit the signal into the cell to create a physiological effect. In membrane receptors, the effector domain may activate enzymes, open or close ion channels, stimulate second messenger systems, or trigger intracellular signaling cascades. For example, G protein coupled receptors can activate cyclic AMP pathways, while ligand-gated ion channels rapidly alter membrane permeability. These mechanisms usually produce fast responses. In intracellular receptors, the effector domain often works differently. These receptors commonly bind to DNA and regulate gene transcription, ultimately influencing protein synthesis. Because gene expression takes time, these responses are generally slower in onset but much longer lasting. Steroid hormone receptors are classic examples of this mechanism. So, the sequence of receptor activation can be summarized very simply. Ligand binding leads to receptor activation, which triggers effector mechanisms, resulting in a cellular response and ultimately a physiological effect. Core properties of receptors. Now, let's talk about the key features that make receptors so special. First, specificity. Receptors are highly specific for their ligands. This means they can distinguish between molecules with very similar structures, allowing for precise control of physiological responses. For example, even small changes in the structure of a drug or hormone can dramatically alter its ability to bind to the receptor and produce a response. Stereoisomers, which are mirror images of each other, may have very different activities because only one fits the receptor properly. Next, affinity. Affinity refers to the strength with which a drug binds to its receptor. A higher affinity means the drug will bind more tightly and effectively, often requiring a lower concentration to achieve an effect. Affinity is measured by the dissociation constant, Kd. A lower Kd indicates greater affinity. How to find the dissociation constant? Kd is the concentration of the drug at which 50% of the receptors are occupied. By using the receptor binding equation, theta equals the concentration of ligand divided by the sum of ligand concentration plus the dissociation constant, where theta means the fraction of receptors that are occupied, means the concentration of the ligand or drug, and Kd is the dissociation constant. This equation shows how receptor occupancy depends on drug concentration. So, if half the receptors are bound, the drug concentration at that point is your dissociation constant. So, how do we actually find the dissociation constant in the lab? There are two main methods. Binding curve method, we measure how much a drug binds at different concentrations. Then we plot a graph, in x-axis, drug concentration, and in y-axis, receptor occupancy. From this curve, we simply locate the point where binding is 50% of the maximum, Bmax. The corresponding concentration is your dissociation constant. Scattered plot method. We transform the data into a linear plot, bound divided by free ligand plotted against bound ligand. In this graph, the slope equals -1 divided by the dissociation constant. Now, let's understand why the slope in a scattered plot is negative. The binding increases while free receptors decrease, causing the BF ratio to fall as B increases. This creates a negative slope. So, the dissociation constant Kd is equal to the negative reciprocal of the slope of the scattered plot. Think of receptor binding as a reversible reaction. Receptor, R, plus ligand, L, forms a receptor-ligand complex, RL. Now, at equilibrium, the rate at which the drug binds equals the rate at which it dissociates. So the system becomes balanced. In chemistry, for any equilibrium system, we define a constant using a simple rule: products divided by reactants. In this case, the product is the receptor-ligand complex, RL. The reactants are free receptor, R, and free ligand, L. So the equilibrium relationship becomes: the dissociation constant is equal to the product of the concentration of free receptors and free ligand divided by the concentration of the receptor-ligand complex. Now moving to the next property, efficacy. Efficacy, also called intrinsic activity, is the ability of a drug-receptor complex to produce a maximal biological response after binding. Even if a drug binds strongly, high affinity, It may not trigger a full response if its efficacy is low. This is why some drugs are classified as partial agonists. They bind but only produce a limited response compared to full agonists. It determines maximum effect, Emax. There's also saturability. Saturability means that the number of receptors available for binding is limited. Once all receptors are occupied by the drug or ligand, no further increase in biological effect can occur, even if more drug is added. This principle helps explain why drug effects reach a plateau at high concentrations. And finally, reversibility. Most drug-receptor interactions are reversible, meaning the drug can detach from the receptor, allowing the system to reset or be regulated. This reversibility is essential for fine-tuning physiological responses and for the safe removal of drugs from the body. However, some drugs form irreversible bonds, leading to prolonged or permanent effects until new receptors are synthesized. So why are receptors so important? First, they determine the selectivity of drug action. That's why one drug affects the heart, while another targets the brain. It all depends on where those receptors are located. Second, receptors determine the intensity of the response. The number and sensitivity of receptors decide how strong the drug effect will be. And finally, receptors help explain side effects and toxicity. If a drug interacts with receptors in unintended tissues, it can produce unwanted effects. To better understand how drugs and receptors interact to produce physiological responses, several scientific theories have been developed. They help us understand why drugs differ in effect, how strong a response will be, and how receptors behave dynamically. Let's start with the simplest receptor model, the occupancy theory. This theory proposes that the magnitude of a drug's effect is directly proportional to the number of receptors occupied by the drug. In simple terms, the more receptors a drug binds to, the stronger the response produced. The theory assumes that every occupied receptor contributes equally to the final effect. This relationship can be written as the effect, E, is proportional to the concentration of the drug receptor complex, Dr. But there's a problem. This theory assumes that all receptors behave the same, and it does not distinguish between binding and activation. So it fails to explain partial agonists and spare receptors. To fix these problems, scientists introduced the affinity-efficacy theory. This theory recognizes that binding and activation are two distinct properties of a drug. In other words, a drug may bind strongly to a receptor, showing high affinity, but still produce only a weak response if it has low efficacy. So, it separates receptor interaction into two steps: binding, which is affinity; activation, which is efficacy. Mathematically, this can be expressed as effect, E, equals alpha, times the concentration of drug receptor complex, where alpha represents intrinsic activity. This means that binding alone is not enough. The drug must also be able to activate the receptor to produce an effect. This idea helps explain different types of drugs. By separating affinity and efficacy, we can understand why. Full agonists have high affinity and high efficacy. Partial agonists have high affinity but low efficacy, and antagonists have high affinity but zero efficacy. The importance of this model is that it explains several key observations in pharmacology. For example, partial agonists can reduce the effect of full agonists by competing for the same receptors. Antagonists can block receptor effects without activating them at all, and two drugs may bind equally well yet produce very different physiological responses because their efficacies differ. Next comes the rate theory. Unlike the occupancy theory, rate theory proposes that the response to a drug does not depend simply on how many receptors are occupied. Instead, it suggests that the effect depends on how quickly the drug binds to and leaves the receptor. In other words, the response is related to the rates of association and dissociation between the drug and receptor. This idea can be represented as effect, E, is proportional to the rate of association minus the rate of dissociation. According to this theory, rapid interaction between the drug and receptor produces greater stimulation. So, The faster the drug repeatedly binds and unbinds, the stronger the response is expected to be. However, there is an important limitation. This theory does not match many real experimental observations and cannot adequately explain several known drug behaviors. Because of this, the rate theory is now considered mainly of historical importance, rather than a model used in modern pharmacology. Now we move to a more realistic idea, the induced fit theory. This theory proposes that receptors are not rigid, fixed structures. Instead, they are flexible and dynamic. According to the induced fit theory, when a drug or ligand approaches the receptor changes its shape to better accommodate the molecule. So rather than the drug fitting perfectly into a pre-formed binding site, the receptor actually adjusts itself during binding. This conformational change creates a tighter and more specific interaction between the drug and receptor. The idea originally came from enzyme catalysis, where substrate binding changes the shape of the enzyme to improve catalytic activity by properly aligning reactive groups and stabilizing the transition state. Similarly, in pharmacology, ligand binding changes receptor conformation and helps trigger receptor activation and signal transduction. This flexibility helps explain why receptors can recognize specific molecules so accurately, even when drugs have very similar structures. The induced fit theory also explains several important concepts in pharmacology, including drug selectivity, structure-activity relationships, and the foundation of modern drug design. Today, this concept is widely accepted because it reflects the dynamic nature of real protein receptors in the body. One of the most important modern concepts in receptor pharmacology is the two-state model. This theory proposes that receptors are not simply turned on or off by drugs. Instead, receptors naturally exist in a dynamic equilibrium between two forms, an inactive state, called R, and an active state, called R star. This equilibrium exists even when no ligand is present. The relationship can be represented as R is in equilibrium with R star. Drugs work by shifting this equilibrium toward one state or the other. Agonists preferentially stabilize the active state, or increasing receptor activation and producing a response. Antagonists generally have no preference for either state. They simply block the receptor without changing the equilibrium significantly. Inverse agonists, however, stabilize the inactive state, R, reducing receptor activity below its normal basal level. This model is extremely important because it explains several observations that older theories could not. For example, it explains basal receptor activity, the idea that some receptors show activity even without any ligand present. It also explains inverse agonism, where certain drugs actively decrease receptor activity rather than simply blocking it. Overall, the two-state model provides a more accurate understanding of why different drugs acting on the same receptor can produce very different physiological effects. For a deeper understanding of GPCR function, we use the ternary complex model. This model explains how receptor signaling occurs through the formation of an active complex involving three key components: a drug, or endogenous ligand, a receptor, and a G protein. When a ligand binds to the receptor, it facilitates the interaction between the receptor and the G protein. This leads to the formation of a three-part signaling unit known as the active ternary complex. Here, three components come together: the drug, the receptor, and the G protein. Together, they form an active signaling complex responsible for initiating and amplifying intracellular responses. This can be represented as the sum of drug, receptor, and G protein in equilibrium with the drug-receptor G protein complex. Importance of the model are: it provides a more detailed explanation of GPCR signaling. It accounts for signal amplification within cells. It explains how the same receptor can produce different responses depending on its interaction with G proteins. Now, let's talk about a very important concept in pharmacology: the concept of spare receptors. The spare receptor concept explains that a drug can produce its maximum effect even when not all receptors are occupied. In other words, The cell does not always need 100% receptor occupancy to generate a full response. Why does this happen? Because cells use something called signal amplification. When a drug activates a receptor, that receptor can trigger multiple downstream signaling events inside the cell. So even activating a small number of receptors may produce a very large biological effect. Think of it like turning on a microphone connected to a powerful speaker system. A small input creates a huge output. This means some receptors remain spare or unused, even when the maximal response has already been achieved. Mechanistically, the key idea is small receptor activation leads to amplified intracellular signaling, which then produces a maximal effect. One of the most important graph concepts related to spare receptors is EC50 is less than Kd. Here Kd represents the drug concentration needed to occupy 50% of receptors. EC50 represents the concentration needed to produce 50% of the maximal effect. In tissues with spare receptors, a drug can produce a large response before half the receptors are occupied. So the effect curve shifts to the left of the binding curve. This means a lower drug concentration is needed to produce an effect than to occupy receptors. Clinically, this tells us that tissues with spare receptors are highly sensitive to drugs. It also means the tissue can continue functioning normally even if some receptors are blocked or destroyed. Another important implication is that high-efficacy drugs require fewer receptors to produce maximal effects. That is why irreversible antagonists may block many receptors before we actually see a reduction in tissue response, because spare receptors are still available. Now, all these receptor theories connect to one important concept: the dose-response relationship. The dose-response relationship explains how the magnitude of a drug's effect changes as the dose increases. In simple terms, as we increase the drug dose, the response usually increases as well. This relationship is commonly represented using a dose-response curve, which provides valuable information about a drug's potency, efficacy, and safety. Understanding these curves helps clinicians determine the optimal therapeutic dose while minimizing adverse effects. One important type is the graded response, where the drug response increases progressively with dose until it reaches a maximum effect, or Emax. This relationship can be described mathematically by the graded dose-response equation. E equals Emax multiplied by D divided by EC50 plus D, where E is observed drug effect, Emax is maximum possible effect, and D is drug concentration. A lower EC50 indicates a more potent drug. Receptor theories are not just theoretical concepts. They have major clinical applications, from understanding drug action to improving patient treatment. First, let's talk about the clinical relevance of dose-response relationships. Dose-response analysis helps us answer two very important questions: how much drug is needed to produce an effect, and what is the maximum effect the drug can produce? These questions directly relate to two key pharmacological concepts: potency, which tells us how much drug is required to produce an effect, and efficacy, which tells us how strong or effective that drug response can be. Understanding this helps clinicians compare different drugs, choose the most appropriate treatment, and adjust doses for individual patients. In simple terms, dose-response relationships help us select the right drug, at the right dose, for the right patient. Next is drug design, one of the most important applications of receptor theories. By understanding how receptors function, pharmaceutical scientists can develop drugs that specifically target certain receptors. This improves treatment outcomes while reducing unwanted side effects. Also, because different tissues may have different receptor subtypes, drugs can be designed with greater precision and selectivity. Third, receptor theories help us understand why different drugs behave differently. For example, why are some drugs partial agonists? Partial agonists bind to receptors but produce only a partial response, even when all receptors are occupied. This can be useful clinically when we want mild stimulation without excessive activation. Another question is, why do antagonists reverse drug effects? Antagonists bind to receptors without activating them. Instead, they block agonists or natural ligands from binding. This is especially important in treating situations like drug overdose or excessive receptor activation, where blocking receptor activity becomes therapeutic. Fourth, receptor theories help explain tolerance. When receptors are continuously stimulated over time, cells may respond by reducing receptor number, called downregulation, or by becoming less responsive. As a result, the same drug dose produces a smaller effect, meaning higher doses may eventually be needed to achieve the same response. Finally, receptor theories help us understand drug interactions, especially different types of antagonism. In competitive antagonism, the antagonist binds reversibly to the same receptor site as the agonist. Because of this, increasing the agonist concentration can overcome the blockade. In contrast, non-competitive antagonists bind at a different site, or bind irreversibly, which reduces the maximum response possible even if more agonist is added. This distinction is clinically important because it influences how drugs are selected and managed in patient care. Let's simplify pharmacology into one powerful sequence. How does a drug actually produce its effect in the body? Let's break down the process of drug action on receptors into clear sequential steps. Step 1. Drug binding. When a drug reaches its target, it binds to a receptor, just like a key fitting into a lock. This is called affinity, the ability of a drug to recognize and attach to its specific receptor. But this binding is not permanent. It happens through weak, non-covalent interactions, like hydrogen bonds, ionic bonds, hydrophobic forces, and van der Waals interactions. These forces are reversible, which means the drug can bind and unbind dynamically. And most importantly, this binding is highly specific. Only the right drug can fit the right receptor. Step 2. Conformational change. Once the drug binds, the receptor doesn't stay the same. It changes its shape. This structural shift is the most critical step because it converts a simple binding event into an active signal. In other words, this is the moment the receptor gets switched on. Step 3. The receptor now activates signaling pathways. This can be explained by modern receptor theories, like the two-state model or the ternary complex model. This can happen in several ways. It may open or close ion channels, activate G proteins, trigger enzymes, or even regulate gene transcription. This step is where the actual biological effect is produced. Step 4. The drug produces its effect. This depends on efficacy, the ability of the drug to actually activate the receptor and generate a response. A drug may bind well, but without efficacy, it won't produce an effect. Step 5. The final response is modified by the body. First, spare receptors. Even a small number of activated receptors can produce a maximum effect. Second, receptor regulation. The body can increase receptors. Stronger response, decrease receptors. Reduced response. Before we move on, let's quickly recap what we've covered so far. We explored what receptors are, their molecular nature and key properties, how they work, and the main theories explaining drug-receptor interactions. We also looked at the importance of receptors for drug action, clinical applications, and concepts like spare receptors and dose-response relationships. Understanding these foundations is crucial for making sense of the different types of receptors and how they influence drug effects. In our next video, we'll explore the different types of receptors, like ion channel receptors, G-protein coupled receptors, enzyme-linked receptors, and intracellular receptors. Don't miss it. Thank you so much for watching. If you found this video helpful, please give it a like, subscribe, and hit the bell so you don't miss our future pharmacology lessons.